So today I'll be presenting on Ambystoma mexicanum, more commonly known as the axolotl, a species that occupies a uniquely important position at the intersection of conservation biology, evolutionary biology, and biomedical research. The axolotl is not simply an interesting-looking salamander. It is a living model organism that has fundamentally shaped our understanding of vertebrate regeneration, developmental biology, and the consequences of extreme habitat loss. At the same time, it is one of the most critically endangered vertebrates on Earth, restricted to a single, heavily degraded lake system in central Mexico. What makes the axolotl particularly compelling from a zoo science and animal management perspective is the tension between its abundance and captivity and its near extinction in the wild. There are hundreds of thousands of axolotls in laboratories and private collections globally, yet the wild populations may number fewer than 100 individuals. This paradox raises important questions about what captive populations can and cannot do for conservation. Questions I'll return to throughout this presentation. We'll move through the species taxonomy, biology, ecology, population status, and the recovery efforts currently underway, framing all of it through the lens of applied conservation science. Let's start with the biology. The axolotl is a neotenic salamander, and neoteny is a key concept here. Neoteny refers to the retention of juvenile or larval morphological traits into sexual maturity. In most amphibians, metamorphosis involves a dramatic transition. The animal absorbs its gills, develops lungs, and transitions to a semi-terrestrial lifestyle. The axolotl, however, bypasses this process entirely. It reaches reproductive maturity while maintaining its larval body plan, including those distinctive feathery external gills, which are used for aquatic respiration throughout its entire life. It's worth noting that the axolotl can be induced to undergo metamorphosis in laboratory settings through hormone treatment, specifically thyroxine. But this never occurs naturally in the wild. This distinction is biologically important. Neoteny in the species isn't simply delayed development. It's an evolutionary fixed trait tied to its permanent aquatic environment. The axolotl also lacks eyelids, which is consistent with its fully aquatic existence, and it ranges in size from approximately 6 to 18 inches as an adult. And then there's the conservation status. Critically endangered on the IUCN Red List, that designation reflects a very specific and severe set of criteria. Population decline exceeding 80% over the past 3 generations, highly restricted range, and ongoing threats with no sign of reversal. The axolotl meets all of those criteria. Taxonomically, the axolotl sits within the class Amphibia, order Caudata, the salamanders, and the family Ambystomatidae, commonly called the mole salamanders. Within its genus Ambystoma, it is most closely related to the tiger salamander complex, Ambystoma tigrinum, and related species. This phylogenetic relationship is actually central to understanding the axolotl's neoteny. Tiger salamanders do undergo metamorphosis, yet they and the axolotl share a recent common ancestor. This tells us that neoteny in the axolotl is a derived trait. It evolved relatively recently in geological time as an adaptation to stable, resource-rich aquatic environments where remaining aquatic was more advantageous than transitioning to land. There's also an interesting genomic angle here. The axolotl has one of the largest known vertebrate genomes, roughly 10 times the size of the human genome. This size has historically made genetic research challenging, though recent advances in sequencing technology have allowed researchers to begin mapping it more completely. The genome's complexity is thought to be related to part of the species' extraordinary regenerative capacity. The axolotl's natural habitat is extremely restricted. It's endemic to a specific lake in Mexico City, and historically also inhabited a nearby lake, which has since been completely drained for urban development. What remains today is not really a lake in the traditional sense. It's a network of canals. The traditionally raised agricultural plots developed by the Aztecs, located in the southern part of Mexico City. The axolotl requires very specific conditions within the environment. It needs muddy substrate that supports burrowing and egg-laying behavior, dense aquatic vegetation for shelter, oxygen-rich water, and cool temperatures, which ranges typically between 14 and 20 degrees Celsius, or roughly 57 to 68 degrees Fahrenheit. The problem is that the lake sits within one of the most densely populated urban areas in the Western Hemisphere. Mexico City has a metropolitan population of over 20 million people, and the pressures of urbanization, agriculture, and human waste have fundamentally altered the chemistry of the water and ecological structure of the axolotl's habitat. What was once a large productive system is now a fragmented, polluted canal network. This extreme endemism, existing in a single, heavily impacted location, is one of the defining features of the axolotl's conservation crisis. The axolotl is a carnivorous predator and functions as a secondary consumer within the food web. Its diet consists primarily of worms, aquatic insects and their larvae, small fish, and crustaceans. Mechanistically, axolotls are suction feeders. They create a rapid negative pressure in the oral cavity, drawing prey in with the surrounding water. This is a passive but highly effective predation strategy that requires relatively little energy expenditure, an advantage in an environment where prey availability can be variable. Their role as predators is ecologically important. By regulating populations of invertebrates and small fish, axolotls help maintain trophic balance within the canal ecosystem. When apex or mesopredators are removed from an ecosystem, the consequences cascade downward, a process well documented in both aquatic and terrestrial environments. The decline of the axolotl therefore has implications, not just for the species itself, but for the broader ecological function of the wetland system. It is also worth noting that in captivity, axolotls are typically fed earthworms, bloodworms, and commercially prepared pellets. Replicating a nutritionally appropriate diet in managed settings is generally achievable, but behavioral enrichment related to natural foraging is more difficult to replicate. I would like to take a moment to highlight a few aspects of axolotl biology that are, frankly, just remarkable. The most scientifically significant is limb regeneration. Axolotls can fully regenerate amputated limbs, including bone, muscle, nerve tissue, and vasculature, without scarring. This is not simply wound healing, it's true epimorphic regeneration involving the formation of a blastoma, a mass of dedifferentiated cells that essentially reprogram themselves to rebuild the lost structure. They can also regenerate portions of the heart, spinal cord, and retina. No other vertebrate does this with the same fidelity and consistency. The axolotl also doesn't undergo natural metamorphosis, as we've discussed previously. And as a model organism, it has been studied in labs for well over a century, one of the longest-running vertebrate research traditions in biology. Finally, its name. Axolotl derives from the language of the Aztecs and is associated with the god of lightning and death who was said to guide souls through the underworld. The species was culturally significant long before it became scientifically important. The axolotl's decline is the result of multiple compounding anthropogenic threats, and it's important to understand them as interacting stressors rather than isolated problems. Habitat destruction is the foundational threat. The draining of the lake and the progressive urbanization of the basin have reduced available habitat by an estimated 90% or more over the past century. What remains is fragmented, and fragmented populations face additional genetic and demographic risks. Water pollution compounds this dramatically. Agricultural runoff introduces nitrates, phosphates, and pesticides into the canal system. Urban wastewater, much of it is only partially treated, raises pathogen loads and depletes dissolved oxygen. Axolotls are highly sensitive to water quality parameters. Even moderate increases in ammonia or nitrate concentrations can be lethal, making individuals more vulnerable to disease. Invasive species represent a third major driver of decline. Tilapia and the common carp were introduced to the basin in the mid-20th century, largely as a food source. Both species prey on axolotl eggs and juveniles, and they compete directly with adults for food resources. In survey data, sites with high tilapia or carp densities consistently show little to no axolotl presence. Invasive species management is therefore inseparable from axolotl recovery. These three threats interact. Habitat loss concentrates the remaining population into smaller areas, where population and invasive species pressure is a lot more intense. It's a classic endangerment feedback loop. Current population estimates for wild axolotls are deeply uncertain, but all estimates are alarming. The most frequently cited figure is fewer than 1,000 individuals. This likely reflects a generous upper bound. Field surveys conducted since 2014 have recorded dramatically declining encounter rates, with some transects finding near zero individuals in areas that previously supported robust populations. Some researchers have estimated effective population sizes in the low hundreds or even lower. This matters for several reasons. Population viability analysis, or PVA, is a modeling tool we use to estimate extinction probability over a time-given demographic and environmental stochasticity. For a population of fewer than 1,000 individuals in a single highly degraded location, the extinction probability over a 100-year horizon is very high without intervention. Meanwhile, the axolotl is one of the most common amphibians in captivity globally. It's an incredibly popular pet, a laboratory standard, and is bred extensively in research institutions. But here's the critical point. Captive populations are not conservation equivalent to wild populations. Most captive axolotls are descended from a very small founding group brought to Europe in the 1800s, meaning they carry limited genetic diversity compared to wild individuals. They also lack behavioral competencies for natural foraging and predator avoidance that would be necessary for successful reintroduction. Captive populations serve a research and educational function, but they cannot substitute for a self-sustaining wild population. Recovery efforts for the axolotl are ongoing and involve a combination of in situ and ex situ strategies. Habitat restoration is one of the most critical long-term interventions. Several NGOs and government agencies, in partnership with the local farmers, have established axolotl sanctuary zones within the basin areas where invasive fish are removed, water quality is monitored, and native vegetation is restored. These provide a template for what a restored ecosystem could look like, and early data suggest axolotl density is higher within the sanctuary zones than in surrounding canals. Captive breeding programs with a conservation orientation as distinct from the commercial pet trade are also underway. These programs aim to maintain genetically diverse populations that could serve as insurance against complete wild extinction and eventually as a source for reintroduction. Reintroduction ecology for amphibians is complex, and success depends heavily on whether the habitat quality has improved sufficiently to support a released population. Water quality improvement is a necessary prerequisite for any of this to succeed. Efforts are being made to reduce agricultural runoff and improve wastewater treatment in the watershed, though progress is slow given the scale of the urban infrastructure involved. It's also worth acknowledging the socioeconomic dimension. The farmers who work the land around the basin are both stakeholders and potential conservation partners. Programs that create economic incentives for habitat stewardship, sometimes called conservation payments or PES schemes, are increasingly seen as essential to long-term success. Conservation that ignores local communities rarely works. The axolotl's survival ultimately depends on whether urban and agricultural pressures on the basin can be meaningfully reversed, a challenge that is as much political and economic as it is biological. To bring this together, the axolotl is a species under extreme pressure from well-documented human-caused threats. Its biology is remarkable. The regenerative capacity alone has generated decades of research with direct applications to human medicine. Its ecological role in the river system where it is found is functionally important, and its conservation situation is urgent. What the axolotl illustrates, perhaps better than almost any other species, is the gap between scientific understanding and conservation outcomes. We know exactly why it's declining. We know what needs to change. The challenge is implementing that change within a complex social, economic, and political landscape. That is ultimately the challenge of applied conservation biology. Not just understanding the species, but navigating the human systems that determine their fate.